We will be starting out with Taylor Cubbage from, she's a master's student at the University of Alaska Fairbanks. And I got to hear her presentation earlier this week, and it's really fascinating, and I'm really happy I get to hear it again and do some more digesting on the really awesome stuff, work she's doing with pike, invasive pike. So Taylor, if you are ready, take it away.
Awesome, thank you, Nicole. Yeah, so today I'll be giving an update on chapter 1 of my master's research. So yeah, kind of shifting gears and thinking about invasive northern pike. Unfortunately, we're all probably pretty familiar with the impacts of invasive pike in Southcentral Alaska, where they've spread throughout the Matsu, Cookina, and Kenai regions. And here they predate heavily on our native fishes and have been implicated in causing the declines of certain salmon runs like the Alexander Creek Chinook salmon. So management agencies are working really hard in this region to do outreach and suppress and eradicate these pike populations. However, because these invasive populations are really close to native populations in Alaska, this provides a really unique opportunity to compare and contrast the traits between the populations to see what might be different and what also might be driving the invasion success of pike in South-Central Alaska to help inform management tactics. So what might be some drivers of invasion success? There's been quite a bit of work trying to characterize traits of successful invaders to predict and prevent future invasions. And a lot of studies have honed in on the trait of high phenotypic plasticity as being associated with invasion success. And this is essentially when an individual has, a constant genotype, but they're able to express a variety of different phenotypes to rapidly adapt to new environments, such as being introduced into new habitats like an invasion. So there's already been quite a bit of work comparing and contrasting phenotypic plasticity between invasive and native pike populations in Alaska, including evidence that there's morphological plasticity and trophic plasticity between the populations. However, metabolic plasticity hasn't really been explored yet. And this is really important in terms of variation in metabolism among individuals because it doesn't only have implications for individual growth, but also population level differences. And so what I mean by that, thinking about metabolic rate, metabolism is of course highly influenced by water temperature in fishes. It's positively associated with it. But studies have shown that individuals with naturally high metabolism have increased exploratory behavior and dispersal tendencies, which of course can be related to invasion success. Additionally, individuals with high metabolism can also have increased food consumption to fuel that metabolic rate. And if there's plenty of food to fuel that high metabolism, then growth rates can increase as well. Additionally, if there's plenty of food and fast growth rates, there's lots of accumulation of lipids and proteins in individuals, which can lead to improved body condition for future life processes like overwinter survival, fecundity, and fitness. And if you have a lot of high individual fitness in the population, this can lead to overall population growth. So you can see how these traits sort of interact together, environmental and physiological. And there's also evidence showing that the process of invasions can actually select for individuals with high metabolic rate to kind of facilitate all of these different processes that improve invasion success. So to see if there are differences in plasticity and metabolic rates between the populations and all these different cascading effects, I decided to compare and contrast these specific traits among populations in Alaska in pike to see if there are differences. So in terms of collecting the fish and experimental design, we collected pike from both invasive and native habitats in Alaska from both river and lake areas to account for potential differences due to habitat type rather than invasive or native status. So we collected 28 invasive river pike from the Deshka, 30 invasive lake pike from Nancy Lake, 34 native lake pike from Minto Flats and Lakes on Eielson Air Force Base, and 31 native river pike from the Tanana and Chena rivers. So in terms of measuring those physiological metrics that I mentioned, that involved euthanizing each of those fish that we collected, taking tissue samples to run metabolic enzyme assays to get estimates for metabolism, collect the cleithra from each pike and age them, and then back calculate lengths to determine growth rates. And then to measure body condition, I used proximate component analysis to get percent lipid of each fish, which is kind of known as energetic status. And then lastly, to take a look at food consumption, I used the diet composition of the pike, just identifying and categorizing the diet items from stomach contents, and I'm also going to be using some stabilized tope analysis as well. And then to kind of characterize the water temperature among the populations, I didn't have the foresight to be able to put out hobo loggers at all these different collection sites. So post hoc, I kind of gathered some summer water temperature data from these different populations to see how those regimes might be different. In terms of data analysis, I just tested for differences in these characteristics among the populations to see where the differences are. And I'm also hoping to use a larger modeling approach, maybe linear mixed effects or principal component analysis, to model growth rate as a function of those other physiological traits, because as I mentioned in that introduction slide, all of these traits are really interconnected. And so hopefully by plotting these different relationships between the populations, we can see what's unique in the invasive populations to kind of figure out what's driving their invasion success. So now getting into the results from this study, looking at the size and age of those populations. This is a fork length distribution of those four populations, and then we've got mean fork length and median age on the right here. So what we saw is that invasive lake pike tended to be the smallest and youngest of all the populations, with invasive river pike being a little bit larger and older. And then native lake pike had sort of this bimodal distribution with quite a few small fish, but quite a few large fish, resulting in the largest mean size and median age estimates. And then native river pike had kind of a more unimodal distribution with more moderately sized and aged fish. So the main takeaway from this plot is that invasive pike tended to be much smaller and younger than native populations, and this definitely makes sense because of years of gill netting and suppression on those invasive populations by management agencies. Something else to note is that, because these population distributions are pretty different, I only had common observations from young individuals across all the populations. So as I go through the next slides comparing those physiological traits, I'm only comparing those traits among the younger fishes from the populations, just to control for potential differences due to size and age. So now I'll kind of walk you through the differences in those environmental and physiological metrics between the populations. First off, looking at mean summer water temperature. So this is water temperature plotted on the y-axis with those different populations on the x. And the main takeaway from this plot that we see is that invasive populations tended to be warmer than native populations. And within a given population, the lake habitats tended to be warmer than river habitats, which definitely makes sense in terms of rivers are often having a lot more diverse and cooler hydrologic inputs. Now looking at basal metabolism estimates from the enzyme assays that I used for a subset of those pike. So what we see here is that, regardless of invasive or native status, we see that invasive populations tended to have higher metabolic, lake populations tended to have higher metabolic rates than those river populations. Then taking a look at the diet composition among the populations. So I identified all the diet items, but I decided to just consolidate it into mean percent fish in the diet because this seemed to explain a lot of the differences here, just looking at mean percent fish. And so what we saw here was that invasive populations have much higher percent fish in the diet relative to native populations. But when I tried to see if there are differences based on habitat type within a given population, the trends weren't significant, but you can definitely see how river populations in the native range tended to have higher percent fish in the diet and lake populations, whereas mean percent fish in the diet was pretty similar between the invasive groups. Lastly, looking at body condition measured as percent lipid content in these young pike. So what we see here is that our invasive populations had significantly higher mean percent lipid content relative to native populations. And we kind of see this trend here where within a given population, taking a look at native fish, river fish tended to have higher lipid content than lake fish. And this trend's sort of visible here in the invasive population, but it wasn't significant. So now kind of taking a step back and thinking about how all those traits kind of combine among the four populations. So breaking it down one by one, looking at native lake pike from the Minto Flats and small lakes in Eielson Air Force Base, we saw pretty warm water temperatures resulting in higher metabolic rates. But because the diet items in these systems were, mostly macroinvertebrates, very few fish in the diet items, we saw pretty small fish with very low growth rates and low lipid accumulation. Now looking at the native river fish from the Tanana and Chena rivers. Here we saw the coldest water temperatures across the populations, resulting in lower metabolic rates. However, these fish had more access to fish in their diet items, so we saw a little bit faster growth rates and more lipid accumulation than native lake pike. Now looking at the invasive river pike from the Deshka. Here we saw warmer water temperatures than the native populations, resulting in higher metabolic rates. And because they had, much more access to this really abundant juvenile salmon and fish prey, we saw really fast growth rates and greater lipid accumulation compared to both of the native populations. Then lastly, looking at our invasive lake pike from Nancy Lake, we saw the warmest water temperatures and high metabolism here, as well as really high fish in the diet, resulting in the fastest growth rates across all the populations and really good lipid accumulation. So now thinking about the implications of this work, we know there's definitely a difference between body condition among the populations. Invasive pike tended to have much greater lipid storage than native populations, which could potentially improve over winter survival, future fecundity, population growth, and dispersal into new habitats. So this might be a really important mechanism for why invasive pike in South Central are so successful. Additionally, we did see metabolic and growth plasticity among the populations. There are definitely differences there, but it seems like it's more a function of temperature and food availability rather than the process of invasion selecting for individuals with high metabolic rates. The only way to really test this would be to use common garden experiments to pool individuals from all these different populations and have them in the same temperature and food availability conditions to really see if invasive populations have naturally higher basal metabolism. So now thinking about management. So we know that these warm lake systems with really abundant food are, hotspots for really fast growth and really improved lipid accumulation. And so these are probably areas that would probably have, the most impacts to salmon consumption in terms of these pike, being optimized for growth and consumption. Additionally, it's important to think that, warming conditions will continue in South Central Alaska and elsewhere. And so as this warming trend shifts to, a thermal optimum for growth in pike, there's probably going to be greater impacts to salmonids through consumption. And with Ben's work with bioenergetics, we'll probably be able to get a more qualitative or quantitative approach to really understand the actual consumption rates of pike on salmonids, now and into the future as temperatures are likely to warm. So with that, I'd like to thank you all for listening and just acknowledge that this work was gratefully conducted on the Lower Tanana, Dene, and Dena'ina lands. And I'd like to thank all the people that have helped me throughout this process and acknowledge that this project is funded by the U.S. Fish and Wildlife Service. And I've also had a bunch of help from the Alaska Department of Fish and Game. And I'm supported academically through UAF and the Alaska Cooperative Fish and Wildlife Research Unit. So thank you for listening.
Thank you so much, Taylor. For those of us working on management of this species in this region, it's really exciting to see this work happening. So thanks for your efforts. We have maybe a minute for questions. You're getting some compliments on your presentation in the in the chat. There was from Bruce said large pike readily prey on small pike. So what might happen if we allow invasive pike to get larger? It looks like Parker Bradley, ADF&G, responded to that, but not to the entire group. So I wanted to share what he said and then give you a chance to respond to that as well. It says, cannibalism is actually very rare in many systems in South Central, even in large pike. Letting them get larger just means they'll produce more eggs, which is definitely something we've seen in the systems in my district. They like to eat salmon before they eat themselves, but yeah, I'd like to see what your thoughts are on that too.
Yeah, I definitely echo what Parker said. I didn't see any cannibalism in the invasive populations that we sampled, even though, I only really did sample smaller fish. And across the native populations, cannibalism was also pretty rare in those larger individuals. So if we really do want to try to stop, initial production of eggs, that's definitely a good place to start, is just not letting them get very big, because those young pike, even though they're still able to produce, they're probably producing a lot less eggs and kind of helping us mitigate the overall process of management for sure.
Great. Julianne O'Doherty, also from Fish and Game, has a question. She says, thanks, Taylor. It was very interesting. I had one question. Are the invasive pike all of Alaska originally, or any evidence there were also introductions from out of the state?
Yeah, so I think the initial introductions to the Matsu area were traced back to an ancestral population in Minto Flats back in the 1950s. But I want to say there were some I think in Chase Jalbert's study looking at the genetics between the different populations, there was sort of this like outgroup that was really unique, and it wasn't really able to be traced back to the native populations. And I don't want to, say that like what state it was, but I do want to say it was maybe from Canada or maybe from the lower 48 even. I think that group was from the Kenai Peninsula, where they found that interesting outgroup.
Okay. Interesting. Well, thanks again. Going to move on to your colleague. Also got to hear from him earlier this week, those of us in the Pike Committee for the Alaskan Invasive Species Partnership.